And the next speaker is Rita Carsetti, and she's talking about the mucosal immunity in children and systemic immunity in adults.
Thank you. Thank you very much for the invitation. And today I'm not talking about my favorite cells because Marika said everything, so I talk about something else. How do I go forward? Yeah. Okay. So, but I start with my favorite picture, so that the memory, the immune memory is composed of cells and antibodies, and we have IgM memory cells, which memory cells, long-lived plasma cells, which Andrea showed to us a long time ago, and we found that those are really important for the maintenance of antibodies long time after vaccination. So in reality, with the COVID, what did we learn? We learned that really the immune response works like this, that you immunize somebody, or this guy has an infection. Then after 2 weeks, you have memory cells, you have plasmablasts, you have all this population that Marika has shown us, they contract, they will diminish, and also the antibodies will go down, and we will have some antibody left because long-lived plasma cells are producing antibodies. The memory cells don't go down, they stay. And we all very happy because they work very well. If you have a recall vaccination or immunization or infection, and we do everything from new, and we probably increase the number also of long-lived plasma cells, at least that has been shown. But you know, it's important that level of antibodies that you have in the middle, you know, when your antibodies go down. One thing we have learned is that in reality, this may be different depending on the infection, because if you have to face meningitis, if you have to fight tetanus, it's really important to have antibodies. And also for very young children, it's very important to have antibodies that work, you know, so very effective antibodies against pertussis and against RSV. And the problem is that tetanus or meningitis will not give you the time to do a response. And the child can't do the response because he doesn't have the memory cells. So sometimes it's really very important for certain disease to have a sufficient level of antibodies. And we don't know how to influence that level of antibodies because we don't know how to induce, for example, more long-lived plasma cells. And for other cases, you know, like even for COVID, in reality, it's not really so important to have a high level of antibodies all the times because we can do a response. We can activate our memory cells, our memory T cells, and who cares? In reality, even pertussis and RSV in the adults do not require a very high level of antibodies because you get cough, but you don't get a severe disease. So these are things we haven't really learned as yet. I don't know why this doesn't go. The other important thing that we have learned from the COVID-19 emergency is that we always forget that most infections start at the mucosal side. And of course, at mucosal side, when we have already experienced the disease, like influenza, for example, we do have some protection because we have the IgA that are exposed on the surface of the epithelial cells. We have antibodies that go from the serum and go to the tissues. And we have memory cells, resident memory cells, or other cells that can migrate, but they have to be memory because the naive cells, of course, stay in the lymph nodes. And if you want to make a response in the germinal center somewhere, you really need 2 weeks, which is too long to be sick. And in fact, this has been demonstrated by the COVID-19 in the patient who got the disease in the beginning, you know, without vaccination, without experience, we didn't have anything. And this was really, you know, complicated. One thing we really have to think that all newborns are in these situations. You know, newborns really have to rely on some antibodies that the mother is giving through the placenta, but really don't have anything. And in reality, for example, for COVID, they did very well. So one important thing is to study, you know, how neonatal immunity works. don't know if you ever got, like measles as an adult or varicella as an adult. I did. And my daughter got it as a child. It was quite different. In adults, it's horrible. The other thing we learned from the COVID experience is that do we have enough antibodies at mucosal level? So I try to find papers to understand how important are antibodies and how many we get at mucosal level. So this is a paper, this was made with monoclonal antibodies against COVID. And you see that having enough antibody matters, because if you treat people with the monoclonal antibodies and the antibody levels decline, then you see, when the antibodies level decline, you really have a severe disease. So it's really important to have the antibody levels. But how many antibodies really go to mucosal side? So I found only this paper, which was very interesting. These were children treated with this nirsevimab. It's an antibody against RSV. All children now in Italy at least get these antibodies. And some of these children, 10 to 30% of the children who got the antibodies still get RSV. So people try to measure how much of the antibodies that you have in the serum goes to your nose. It's 1 to 1,000. So of the antibodies that we have in the blood, only 1,000 of those is going to go to the mucosal side. And then, of course, if you have the antibody that are low, like when you are far away from the immunization, it's 1,000 of very low. So that's why we are not protected. And that's why, protection is very good when you measure like 2 weeks after the immunization. So we really have to learn more about this. And we have to learn more, I think, about mucosal immunity of the respiratory tract, because most of the things we know are about the gut, where it's easy to get biopsies, but the gut has its own immune system, really. It's quite different. About long-lived plasma cells, this is something we are just starting. Actually, I have to talk to Andreas about this. So these are the famous long-lived plasma cells in the bone marrow. So in the bone marrow of an adult, you can find cells which are plasma cells. They express CD138. They're also all gating, but these are all plasma cells. These are still CD19 positive. This is the CD19 negative population. You really find those cells in the adults. And actually, I started to look at this because there was this paper in Nature Medicine saying that we do not get long-lived plasma cells from SARS-CoV-2 immunization. And I was very curious because we don't really know how plasma cells from the CD19 positive group, but downregulated CD19 and becomes really long-lived. We don't know. In the mouse, that's been shown that it takes time, but what happens in this time, nobody really knows. So we started to look at the long-lived plasma cells in children, and I have some samples from children who are bone marrow donors for their siblings. Look what is in children. This is 8 years and 14 years. So really don't have the long-lived plasma cells. So that's why I think that in reality, this paper in Nature Medicine really tells us that 2 years after you don't have them. But we don't really know what are the rules and if we will get them. So now we want to look at this population in more detail. It's very difficult because you have to have enough cells, and that's the main problem that we have right now. We're trying. The other thing we learned from the COVID-19 is this data on the fact that we have a stereotyped response. Look at this one here, the mRNA vaccine. You have a strong response. You make a lot of memory cells, lots of antibodies, and then you get Omicron, and you don't really make cells that are specific for Omicron because your immune response is imprinted, and you use the same cells that you develop for the response to the wild type virus. Sorry, I'm really sorry about this. So if you see this is a study, it's really interesting. It's from the Richard Howe group in Beijing, they look in people in China who got the inactivated vaccine. These guys didn't have any problem. They could respond also to Omicron. So they didn't have a very strong response, but they could respond to Omicron. Then he also immunized mice, and he saw that also mice don't get imprinting. They respond to Omicron. And so he sorted the cells from the people who were vaccinated with the mRNA vaccine, and he demonstrated that there was a special VH gene, which is used by high affinity memory B cells in humans, and this 353 or 366 family. And this is the problem. This is very dominant, this type of VH, and we always use that. So what he did, he made even knock-in mice with 353, and then also in this case, the mice really show a stereotype response, like they have an imprinting and they are not really able to make cells that recognize Omicron. So this looks very scary, but then we looked at children that were either vaccinated only or vaccinated and then got an infection, or children were never vaccinated, never had an infection, but then they got Omicron. And we asked, what happened to their response? First of all, none of the children had the severe disease. It was really not a severe disease. These are the memory cells. So now, if you look here, these are the vaccinated only. They make a lot of memory cells specific for wild type virus. Also, the guys that are vaccinated and infected, they make these cells. The guys who only got the Omicron infection don't see the wild type virus because they never saw the wild type virus. They only don't know what happened. Sorry, excuse me. I always do this. I'm really stupid. I'm sorry. These are vaccinated and then infected. They still see very well the wild type, and the Omicron only don't see the wild type because they never saw it before. They were not immunized. So then there are cells that bind at the same type of wild type, but also Omicron, and you can see that everybody has them besides the guy who only saw Omicron for the first time. And then here, in the children who got the Omicron infection, you really have the Omicron-only population. Now we want to sequence these cells. It's again difficult because you have few cells to see whether they use a different VH genes than the one used by the other children. Antibodies level are not a problem. Even of course, with the limit of measuring neutralizing antibodies, everybody is able to neutralize the wild type. Of course, the Omicron-only people less, but they all neutralize Omicron. So probably in all of these things we learn about the stereotype response, also about the imprinting is true, but in reality, our response is so polyclonal that it really is not affecting so much our ability to respond to the infection. And now this is going back to really very young children, because I'm telling you about a small study that we did during the COVID time, because we had the chance of studying mothers and children, and these were the mothers who were found to have COVID at the time when they were delivering the child. And we did this interesting study. So the baby, of course, doesn't have his own memory cells, doesn't have any preformed response. So he has an innate response, an adaptive response. The innate response is mostly anti-inflammatory, which is very good because the newborn is going to be exposed to a lot of pathogens and also commensal that could really elicit very strong responses. And the adaptive response, he has to create them. Doesn't have any. So he has to rely on the antibodies that the mother is going to give through the placenta and with the breast milk. And the antibodies that you give through the placenta, that happens in the third trimester, and are all the antibodies which we have in the serum, but all of IgG type. So the mother transfers systemic immunity to the baby, and the baby is going to have the same concentration of IgG as the mother. So it's very, very, very efficient. After the baby is born, the mother is going to give antibodies through the breast milk, and it's mainly IgA, very little IgG. And the IgA antibody that the mother is giving are coming mostly from mucosal side, because it has been demonstrated that the cells migrate from the gut and also the respiratory tree to the breast, and then you make IgA for the baby to defend his mucosal side with antibodies that are coming from mucosal side. So it's really genius, I think. And so we did the study because, of course, SARS could have been dangerous at the beginning. We didn't really know for children. And we wanted to see whether the antibody from the mother go to the child, and if the mother is vaccinated, not vaccinated, and so on. So we had children from mothers who were infected at the time of birth. So we got the blood and 5 days after the infection. We also got the saliva and the breast milk, and 2 months after the infection. And then these were women who were vaccinated during the 3rd trimester, at the beginning of the 3rd trimester, and some women were vaccinated against COVID during lactation. And we found what you expected in reality. So this was not very exciting, but these are the numbers. So what we found is that the mothers who had the infection, if you analyze them in the beginning, you know, of course, they don't have IgG because they didn't have time. They were just infected. If you look at 2 months after the baby's born, of course, they had some time to make an immune reaction, so they do have IgG. And these are the people who were vaccinated. Of course, they have IgG. IgA, it's low. In all cases, it's a bit higher, and this one vaccinated during lactation. We don't really know why. The babies. So the babies of the mother who didn't have IgG didn't get any IgG, and also the mother was infected at the time of the birth, so they didn't have time to give antibody through the placenta. The one that were 2 months later, 2 months later, the mama had antibodies in the blood, but the child was already born, so he could not get these antibodies from the placenta. And then these are the children of the mothers who were vaccinated during pregnancies. Of course, they get antibodies. That's all clear, no? That's all normal. And in fact, there is a direct relationship between the antibodies in the baby and in the mother. It's all logic. Then we also look at the antibodies in the breast milk of the mother. And then you can see this is the IgG. You don't get any antibodies really in the breast milk of the mother who had the infection, IgG type. And you get some IgGs in the breast milk of the mothers who are vaccinated. IgA, you see, is not influenced really very much by vaccination. In reality, the thing that is more strange is that the mothers who were infected, so we analyzed them 2 days after we had done the diagnosis, they really had already some IgA in the breast milk. So it doesn't correlate. So immunization during pregnancy, of course, helps. And the mother was infected in the prepartum, they can only protect the child through the breast milk, where they have some IgA, not IgG. Then we had a very strange result, which we didn't know how to explain, was that when we took the saliva of the children at 2 months, so this was 2 months later, only one of the children was infected, this guy here, he was infected after the delivery when they went home. Actually, it's amazing that none of these children ever had a positive swab, never. No, none of them, although they were breastfed from the mother. And these are the children who got breast milk. And then you can see that they had IgA in the saliva. So this is not IgA from the mother, because we were really careful to take the IgA, the saliva like before the meal. So before 4 hours after the last meal. And we could not really explain that. And we look again at everything. And then again, this is the maternal spike-specific IgG in the serum. And we have shown before now that the antibody increase in the serum 2 weeks after. But children, 2 months, sorry, children don't really have the antibody, because besides the guy who was infected, you know, they didn't have the infection. And these are the breast milk antibody, the breast milk antibody response of the mother. This is total IgA, total IgG, and you see that the specific one are only IgA, they're not real IgG, what you have in the breast milk. This is the saliva of the infants, and it's really very clear that these guys do have in the saliva of the infants, so you have, this is total IgA that increases because the baby makes its own IgA and makes also specific IgA, a bit specific IgG, not specific IgM. So we thought, how is this possible? We look in the milk, we look if we find a spike protein in the milk, like the virus. We look at everything, we didn't find anything. And then we said, but what about if the mother is going to give like a spike protein complex with her own antibodies? Maybe antibodies, immune complexes are immunogenic. So what we did, we developed an in-house ELISA where we use an antibodies against spike to coat the plate. Then we put the breast milk in different dilution, and then we develop either with anti-IgA or anti-IgG. And what we saw is that there were immune complexes of IgA and spike in the beginning, when the mother was infected, and then these immune complexes were made only by IgA, and they were disappearing after 2 months. So they were much less. Here you can see this. We did many of these experiments. We did it also in a separate cohorts. So somehow we thought that this means that the maternal milk does passive protection, but maybe is also able to start to trigger some mucosal responses in the children. We are now, for a long time we couldn't do it, but now we are trying to investigate what are the other immune complexes in the maternal milk, because, for example, also the maternal milk contains CMV, for example, and children don't get sick with CMV, although most of the mothers do have CMV in the milk. So we want to see whether this is a more general mechanism. And now, just to finish one more thing, I just want to say something about resting memory B cells. You heard today. And many years ago, when I was still in Germany, we demonstrated for the first time that memory cells are stored in the spleen. So we asked the question whether if I immunize somebody against SARS-CoV-2, but this guy doesn't have the spleen, what happens? Can he maintain this immune response? So we had a group of people who were splenectomized either because they had thalassemia, or they had surgery, or they were born without spleen. In our hospital, there is a center of reference for asplenia. So we immunize these people with the normal mRNA vaccine, and then we measure the first response. I'm not showing that they respond. They have no problem because I immunize them in the arm. They have lymph nodes and they make a normal immune response. Our question was, what happened 1 year after the last immunization? So if I look at serum antibody, each one of them independently of which is the cause of, these are the controls in black. This is, I don't remember this is surgery, I think, congenital and thalassemia, congenital asplenia and thalassemia. And you see that the antibodies, these are neutralizing antibody against wild type, against Omicron. These are children that were splenectomized because of spherocytosis of thalassemia, black the controls, age matched. No problem with antibodies. But now, if you look at the memory cells, that's interesting, because now let's just look at the high affinity cells. These are the controls. These guys all had high affinity cells, 1 year before. 1 year later, none of them has them. They have some activated cells still, but less than the controls. But the resting memory cells are really gone. These are against wild type, these are against wild type and Omicron. We also look at the Omicron only. They're all gone if you don't have the spleen. So what happens? I should demonstrate that these cells were in the spleen. Of course, I can demonstrate it in these guys because these guys were vaccinated after they didn't have the spleen anymore. But we had the spleens from organ donors, and we collect the spleens since a long time. So we had spleens collected before 2019 or after 2021. And we look at the spike-specific population, and you can find them only in the cell, on the spleens of people who were vaccinated, but we got the spleen after 2021. And then we look whether they were in the CD21 high. This is now for B-cell freaks, CD21 high or CD21 low population, and it looks they are everywhere. And this means that the cells you induce in your germinal center, you immunize in the arm, they in reality from the blood go to the spleen, and then probably the spleen act as a reservoir that refuel the peripheral blood with your resting memory cells that can go recirculated and be used everywhere. So I'm finished, really. These are all the people from whom the lab down, and then all the people who gave me a lot of samples. I also got the samples from the healthcare workers of the hospital, and I have to remind you to come to the iOS conference in Vienna because, because I have to make this advertisement, and you should come.
Thank you, Rita. Thank you, Rita. Yes, Andreas, you can start.
Yeah, just a comment to the CD19 positive and negative plasma cells and this confused paper of Nina San's group that you mentioned. Because actually this dichotomy was discovered about 10 years ago here actually at the Charité. And it was obvious that the measles-specific plasma cells, for example, were likely CD19 negative, while tetanus ones were mostly CD19 positive. And that led to the emotional concept actually that the long-lived ones are CD19 negative, the other ones are CD19 positive, without any measurement of their lifetime. Yeah. So now, actually, last year we published what is actually the reason for this difference, and the reason is very simple. That actually plasma cells that come from activated B cells that have seen interleukin-21 downregulate CD19.
Okay.
If they didn't see interleukin-21, they remain upregulated CD4, CD19. Or in other words, the CD19 negative plasma cells come from follicular immune reactions, and the CD19 positive ones from extrafollicular reactivation of memory B cells. So there's actually no reason to believe that one or the other is more short or long-lived. Also, no data actually showing that. And one argument in the case of SARS-CoV-2 would be that the antibody levels against the spike protein of SARS-CoV-1, 2003, when compared between 2003-4 and 2020, remained absolutely constant. Yeah? So there was no drop actually over 17 years in the patients that had been infected. So I think the claim that the SARS-CoV-2 specific antibody secreting cells are short-lived is not substantiated.
That's why we want to do the study, because we think also the same, and that is no demonstration at all.
Sorry for that. So now questions or other comments?
No.
I had a question for here. For the transfer of the antibody antigen complexes through the breast milk and then immunizing the child. Is it something that's spike specific? Do you see that with other? I mean, the other maternal immunizations, for example, now against RSV or pneumococcus, for example. So is it something more generalized or something specific?
That's what we are studying now, to see if it is more general, and it is, but it will not happen with vaccination only if the mother is infected. Because I didn't have time to show the data, but if you look in the women who were immunized, there was nothing in the milk, only in the one who got the infections that we did. So it was so boring, not bad.
It was so clear. Christina.
No.
Chris Falk.
So you just mentioned that the infection was, the mothers were infected, and then you have the immune complexes. What is your idea how it gets there? Maybe I missed it, but I was just thinking, who's schlepping around the spike complexes, so they end up in the mother's.
You know, in the breast milk, the cells go. There are cells that migrate to the breast milk, and they come from mucosal side. So I think that might be the mechanism. Yeah. Because it has been really clearly demonstrated those cells are coming from the mucosal side. And the mother does have antibodies in the mucosal side, no? And cells. Okay. So,
I have one more question.
Yeah.
Actually. So you didn't really talk about the cellular potency of the infants, I mean, around very young children. So is there composition of, let's say, the relation between B1 and B2 cells? Is that the same?
Yeah, I mean, B1 and B2 cells are very difficult to decide, to describe in human. Actually, we studied the B cells in these kids here, this particular one, because it was really interesting. I don't think anybody could do it really, that we had the child from the blood from a child at 5 days, and then we had the same child at 2 months. And you really see the maturation of their immune system, and they have essentially IgM memory cells. All IgM memory.
Okay, final short question, Andre.
In the case of immune complexes, if they have already spikes, so these antibodies are not protective in children, in the saliva of children.
None of these children caught the disease. And they never had a positive swab, although they were staying at home with the mother, and the mother was either breastfeeding them or feeding them with the bottles. Actually, we were totally amazed. We really tested them all the times, and only one got sick. And actually, a funny part is that the husbands got sick. So it's really strange. You know, the children are very good against this particular disease. But you know, everything is new for a child, so you have to be really good.
All right, we close with that.